without further ado, I would like to welcome our first speaker for today, Ryan Smith, founder at True Diagnostics, who again will be presenting on the topic of his company's Dunedin Pace Epigenetic Aging Clock. Ryan, it's a pleasure to have you with us. The floor is yours.
Yeah, thank you all so much for having me. I'm excited to talk today in a great line of speakers. What I really want to talk about today is just a little bit of an introduction to sort of who we are at True Diagnostic. And then in particular, I think one of the best aging algorithms that exist out there in terms of reading DNA methylation for aging-related appearance, and that is that Dunedin Pace of Aging metric. And so just to give you a little bit of background on us, we're a relatively new company. We started offering commercial methylation epigenetic testing and reporting in July of 2020. However, since then, we've developed one of the largest private epigenetic databases in the world with over 15,000 patients with multiple very exciting covariates like full-scale proteomics, full-scale metabolomics, and a wide variety of phenotypes and other lab testing. We're really excited to use that type of data, that large-scale data, to then create new and better ways to interpret the epigenome of every individual, and not just for necessarily longevity-related concerns, but things even beyond that, by looking at things like Alzheimer's, the imprintome, newborn screening, and et cetera. Currently, though, our focus is definitely aging. We have over 30 clinically approved trials at the moment where we're investigating the effects of several types of interventions and looking to create new helpful aging-related algorithms, such as senescent burden level detectors, among others. And we do a lot of that work with researchers as well. So if anyone ever has any interest in doing any specific methylation research, I would highly encourage anyone to reach out to us as we're definitely investing into this field. In addition to that, our commercial offering is an at-home blood test where we will take sort of a fingerstick lancet. We'll run an epigenetic analysis of over 900,000 CPGs and then report that out mainly in age-related reports. And so our age-related reports encompass a little bit of everything. They encompass things like telomere length, immune cell subsets. So you can look at things like the percentages of each immune cells, like your CD4 and CD8 T cell ratio. We do the overall biological aging, separate that between intrinsic and extrinsic aging. And we do this pace of aging, which will be the main point of topic today. As I mentioned, the way that we're able to create some of these new and innovative algorithms is by a lot of that covariate data we're able to collect from our physician group. It's really, I would say, our one sort of secret way to really push the field of aging diagnostics further by gathering a lot of covariate information from all the physicians who are using our testing. And we are particularly, I would say, interested in looking at these multi-omic related values. So looking from everything from genomics and epigenetics to transcriptomics to proteomics, metabolomics, and ultimately those phenotypes we're trying to predict. And so that is the approach that we're taking, and hope to have several multi-omic oriented aging clocks out in the middle of next year. And so really the algorithm I want to talk about today, and one I think is most exciting for our purposes, is this Dunedin Pace of Aging metric. And so unlike traditional biological clocks, this is not reading out an overall age. What it's reading out is essentially your instantaneous rate of aging. So at the time of testing, how fast are you aging? And this has several benefits we'll discuss today as we go along, but we'll be answering sort of all of these different questions. And so before I go into the actual algorithm, one of the things I really want to talk about is how these algorithms are created or trained. Really in the first generation clocks, which were made popular by Dr. Steve Horvath in his 2013 work, what they were sort of training against or predicting against was chronologic age. So they were taking all these large scale methylation data sets in order to predict the chronological age. And that was a really important step, and one that I think is fundamental for the aging research we're doing now. However, these algorithms over time changed a little bit to these second generation algorithms. And these were not trained to predict chronological age. What these were trained to do were to predict markers of morbidity and mortality, so clinical biomarkers. And this is a lot more helpful in the medical space, because instead of just trying to predict a chronological age, if you usually wanted that from a patient, you could typically ask. However, what we're really trying to do is project how rates of aging might affect risk of disease. And these became a lot better with these second generation clocks. However, now this sort of, after sort of advancing from these biological clocks like PhenoAge and GrimAge, the health and retirement study done at USC sort of listed this Dunedin Pace of Aging as a third generation clock. And the reason being is this has been trained on the longitudinal cohort and is able to capture a more instantaneous picture of aging. And so in order to give a little bit of background on this, it's important to know some of the players involved. This was mainly developed by Duke University, and it really started with a cohort from the University of Otago in New Zealand. Really, when Duke first applied for funding for this, they were hoping to look at a cohort and measure their aging throughout an entire lifespan. And actually, when they first asked the National Institute of Aging for financing on this, they were sort of laughed at. They thought that the National Institute of Aging thought that there would be no variation in aging rates in younger individuals, and if they did, it would not be significant to the phenotypes that they would develop later on. New Zealanders who were born in 1972 and were 3 years of age in 1975 when the cohort started. And in this cohort, they measured a lot of really interesting age-related phenotypic biomarkers. They measured everything from postural hypertension to DEXA imaging scans. They did auditory testing. They did markers and measurements of physical function, such as 30-second chair stands. They did even optic nerve scans, as well as dental imaging and even advanced MRIs. And so some of the data that was used in this cohort is really advanced data sets. And again, this is on a longitudinal patient population. So they started with when they were 3, and now these patients are over age 45. And so with all of that data, they sort of defined this 4-step approach. The first was to collect this longitudinal data, then to second, create a pace of aging based on those clinical biomarkers. And with that pace of aging, they then were able to create a methylation-related diagnostic via elastic net regression modeling, which is the method used for most of these methylation-related algorithms. And so you can see here, these are the 19 different blood-based biomarkers that were used to develop this phenotypic aging score. And this is sort of how the mathematical process they use to create that score. And so as you can see here on this x-axis in the graph, to the left, you have little to no biological aging. And as you get further on that x-axis, you would have faster chronological aging. And some of the patients they looked at in this cohort were aging up to 2 years per chronological year. So over 20 years, they might have aged 40 years biologically in this cohort. And so being able to track those differences in aging were fundamental to develop this pathway, which ultimately led to a DNA methylation algorithm which could predict the aging rate in individuals and predict phenotypes outcomes as well. So once they developed this, the idea was how would they validate the Dunedin pace algorithm in different cohorts? And so they looked at several different studies to do this. However, I really want to talk about some of their results at age 45 here in to sort of a way to illustrate how clinically relevant this algorithm already is today. So some of the things I like to show here are some of their performances at age 45, sort of segmented by their rates of aging. One of the things I think is constantly a battle that we have to fight in this longevity space is sort of this false dichotomy between health span and quality of life and overall longevity. I think a lot of people equate older lifespans to poorer quality of life, but these things are not mutually exclusive. And I think that this is some of the phenotypic testing which proves that. If you look out here on this x-axis, you can see the rate of aging. And as you can see, the performance on balance testing was significantly worse with the faster aging. And this type of arrangement did not just happen with balance or physical performance testing. It also happened with things like grip strength or muscle mass and body mass sort of metrics like frailty. It even extended to looking at cognitive decline, where as people who were aging at a rate of 2 biological years per year were on average around 5 IQ points lower than those who were not. In addition to that, we saw a lot of changes in MRIs of the brain as well, where we saw cortical thickness diminished and surface area diminished in older individuals as well. And then lastly, we can even see this on facial aging appearance. And this is one of my favorite things to sort of show, because a lot of times when we talk about aging, we talk about relative risk, and relative risk can be a little bit hard to motivate some patients. And so a lot of times we like to show this picture as well. So these are facial generated images of everyone in the cohort at age 45. On the left, you have the 10 slowest aging members of the cohort as detected by this Dunedin pace algorithm. And on the right, you have the 10 fastest aging members of the cohort. And so what you can see here is that all these people are the exact same age. The difference between the youngest and oldest members of the cohort looks to be a 20-year difference, at least visually. And the way that this is true is that, in fact, they probably have aged that much more due to their faster rates of biological aging. They might have been aging at a rate of 2 biological years per year, while the other people in the group, the slowest aging members, might have been averaging right around a 0.6 biological biological age per year appearance. And so what this goes to show us is that the faster that we're aging, the worse our performance on all of these phenotypic measures, and including facial aging, meaning that we're aging from the inside out, and that age is a predominant factor driving all of the development of these diseases. And so this is a really, really great clock because it's able to predict and pick up a lot of those phenotypic comparisons. But in addition to that, it has some other benefits compared to some of the other clocks. So one of the things we like to point out in comparison to the other clocks, and this is a published paper from that same group at Duke, shows that there's not a whole lot of overlap between some of the other age algorithms, particularly the chronological age or biological trained algorithms, which suggests that this is measuring a different process of aging than what might be captured via some of those clocks. In addition to that, this clock is really exciting because it has a relatively low standard deviation, meaning that we can get very, very precise measurements. This is definitely even highlighted a little bit further by looking at something called the Intraclass Correlation, or the ICC values. And really, to define an ICC value, it is essentially how correlated are one result to the other. So if we took the exact same sample and we measured it twice, how correlated would those results be? And obviously you want a result that's as close to one. You want it to be almost identical to if you're taking the same sample. And so here you can look at some of the most popular aging algorithms and their relative ICC values. You can see with the 2013 Horvath clock, for instance, it is reading right around a 0.675 ICC value. And the beauty of this particular Dunedin pace algorithm is that it is substantially higher, and the only one of these tests which has a high level of precision with an ICC value over 0.9. This is changing, and thankfully, one of the biggest problems with these methylation-related age diagnostics is their variability and some of the noise you might see in those algorithms. And so the Dunedin pace definitely has the best ICC value of any published algorithm to date. However, some of the work that Morgan Levine is doing at Yale with Albert Higgins-Chin is also very exciting, as they're using principal component analysis in order to make the variability of all of these aging metrics significantly lower. But right now, this Dunedin pace represents one of the most precise measurements, which makes it great for personalized medicine. What we can see in some of our patients and our physicians who are using this testing is that they might have the same treatment protocol for two different patients, but each of those patients might have vastly different responses to their rates of aging through this measurement. And so you can get a really good idea of what is working for you to get this as low as possible. One of the other really exciting things about this is that we're also seeing that it works and performs very, very well in saliva as well as blood, which will be able to sort of roll out a saliva-based method of pace of aging detection, which will be relatively low cost and highly precise. And so if we look at the benefits of this algorithm, by far the number one benefit is the precision. Compared to a lot of these other aging algorithms, we can see even statistically significant interventional results in a period of 3 months, where that was very, very difficult and required very large sample size with some of the other age algorithms. In addition to that, it's detecting instantaneous rates of aging. So if, for instance, if someone has lived a very poor quality of life from a health perspective previously, and then they try and change their lifestyle to have optimal aging, we can actually see that change happen rather than still detecting some of their other previous behaviors. And again, this is an algorithm that's been trained longitudinally off the same patients, and so it's not confounded by generational exposures like the prevalence of antibiotics or even leaded gasoline, for instance. And so this represents several features of these aging clocks where there's some significant and vast improvements. And in addition to that, we know that this is incredibly predictive. We already talked about all of those phenotypes which it influences in a positive or negative way. If you can get this below 1 and keep this below 1, it is very, very good for your overall health. Even if you're slightly above 1, even if you're aging at a 1.01 biological per year basis, you would actually increase your risk of death over the next 7 years by 56%, and you increase your risk of a chronic disease diagnosis by 54%. So these risks are very, very predictive and give you a great target to try and keep this below 1 as much as possible. So with that being said, that's a little bit about the algorithm, but I also want to talk about a little about what it's the newest version. This Dunedin pace only came out in the beginning of 2021, although we're definitely already already using it in several investigations to find out a little bit more. And so the first thing that I should say that it has taught us is that the impact of early life is incredibly important. Sort of now, the sort of innovative thought process in aging is that early life adversity can affect this rate of aging, which then proceeds and might predispose people to worse aging-related phenotypes over time. For instance, even measurements of brain function at age 3 in this cohort were predictive of aging-related outcomes at age 45, meaning that aging rates can be set in early life and in utero. And this is backed up by several other studies as well. Particularly, we have looked at this study, which shows that the Dunedin pace is accelerated in early individuals who have experienced early life adversity. So for instance, socioeconomic class is highly correlated with more affluent populations scoring better and having slower aging rates in this metric. And we can actually even detect polyvictimization in this as well, which would then negatively impact that rate of aging. And in addition to that, we see this correlated and corroborated with another study, which looks at the Dunedin pace as it relates to neighborhood level or family level socioeconomic class, where lower socioeconomic groups have worse rates of aging, which sort of lead into this idea that our aging rates are set even at very young ages, and that aging should not be something to consider at 40 or 50 years of age, but throughout the entire life process. In addition to that, this is able to pick up, I would say, socioeconomic disadvantage in a way that no other clock is as well, which provides some additional benefit there too. And I know we're running a little bit tight on time, so I'm going to go through these next slides a little bit quickly. But we also know that there are some validated interventions to change this metric. That's sort of the number one question we get asked is, what do we know about what has been published to change this metric? One of the things that we know based on this calorie study is that caloric restriction, particularly 25% caloric restriction over 2 years, can vastly change these rates of aging. As you can compare the non-caloric restricted group on the left in that graph, and then the caloric restricted group on the right, and their relative change in biological age over the course of 2 years. In addition to that, we also know that the Dunedin pace, or as you can see it here, the Dunedin POAM4X, is by far the most sensitive at actually looking at the effects of these interventions. So it was actually able to capture the anti-aging effect of caloric restriction better than any other age algorithm. And I think this is true for most scientific research investigations. In addition to being a great tool for personalized medicine, it's also a great tool to incorporate into research trials to try and look definitively at what is having some of the best rate and change in some of these measurements. And so, we've also then applied this to things like cognitive dysfunction, where this is best at predicting and picking up changes in cognitive dysfunction or mild cognitive impairment or dementia. And it performs a little bit slightly better than GrimAge on that as well, which has typically been the best. And we've also applied this to things like COVID-19 and mRNA-based vaccines, where we've seen some very interesting results, which I probably don't have time to talk about today. But for instance, we have seen that people over 50 with COVID infection tend to age worse, and people under 50 probably have a hormetic response due to their naive T cells, which actually confers an anti-aging response. And we also see significantly shorter telomeres as measured through our telomere length estimators through methylation as well. We've even applied this data to dasatinib and quercetin related senolytic applications, and had great results at protocol over the course of 6 months, reversals in every age-related metric. And we're applying this even beyond this to several other clinical trials, which you can see listed here, and really hope to report on how this metric is changing the rates of aging, and really what we can look at from population level scales to make recommendations to really affect the phenotypes of aging. And so I know that was a lot of information, but would love to answer it up, open it up for any other questions I might be able to answer.
Thanks so much, Ryan. Very interesting content indeed. For anybody who has follow-up questions for Ryan, I'd ask you to pop those into the Q&A box at the bottom of the screen, and we'll do our best to address them during the panel discussion later today.